Yes. So, the next topic in our discussion is gene therapy. We have seen so far about various things which are associated with gene. What is gene? How gene is expressed? How gene is processed? Then what we have seen is what do you mean by gene uh, cloning? What is gene mapping? So we have seen various aspects which are related to gene, and one by one, step by step, we went for the details about so gene expression. We have seen transcription, we have seen protein synthesis, we have seen various factors. So this genetic information, now we are reaching towards a next important place where we are going to have about what is the gene therapy. Now, so far what we have seen is gene is there. Each person is having, or each organism rather, is having its own genetic makeup. Gene has got one or more function, and by that virtue, this DNA is gene is actually portion of DNA, and from that we can have RNA. From that, when we call it as a gene expression, from DNA to RNA, RNA to protein. So this whole gene expression gives rise to some protein, and because of this protein, it can be structural protein or it can be functional protein. And because of that, these proteins, as they are important, they are structural functional units. They gives variety of activities. So there are more than 1 lakh gene, or even more than 50,000 gene, if they are there. If one gene is responsible for about 2 to 5, these are minimum functions, then how many functions they are carrying? And we can understand that that means these all are functions which are important. Many maybe we are not noticing, but still they are important, they are happening. So this is a regulator machine for RNA production, from chromosome to gene, DNA, RNA transcription, proteins are there, and as I told you, structural and functional activities they are doing. And there are many activities they are making. They can acting as a spacer, they are regulatory proteins, coding, spacer. These are all DNA activities we can see which are there on the chromosome. So organisms like human being can have around 10,000, 10 raised to 5 genetically controlled functions. You can imagine the number. And these are present in the variety of ways, and the outcome is there. That is around various things you will see, that is around 2 meter of the person, suppose overall all area, and there are 2 millimeters of tissue. In that you can see that functionally, if we will go for, then there are the areas which are very small where the cells are there. And if we'll still go into molecular layer, then again the size is very less, that is even 0.001 mm. And these are all responsible, they are all the functional unit of the cell. Now if we'll see various activities, just a modified view we are having. So that is, gene is a DNA, it is going for a protein, and if the gene is transferred, we have seen it in cloning and we have seen during mapping, that this gene is transferred, then it will cause transfer of new function, or transfer of restoring the function, or transfer of interfering of the function. So this thing we can do, and because of that, if we modify anything in the gene, the outcome will be changes in the protein. So example is you can have it, that is the gene is responsible for a function. If gene is having suppose coding for OK, then it is there. If you modify it, it will become KO instead of OK. If gene is transferred, then the function will be transferred from one gene to other gene. And there are many examples are there which are diseases which are inheritable, or which are because of the genetic diseases. There are many effects, and example of these are diabetes, hyperuricemia, glaucoma, then dysplasia, hypercholesterolemia. Many diseases are there. So there are many genes are there, this gene can be again, these defects can be monogenic, or monogenic means from only one inheritance. We know that from parents we get it. So from that we get it. And these genes are there, sometimes they are dormant, and because of the behavior, because of the environment, because of other ambience, there are changes which are there and they can then come. So every person bear one or more latent genetic defects. Each and every person, you, me, everyone is having one or other defect. Many defects are not manifested, and they are not leading to any sign symptoms. But as I told you, there are impact of environment, impact of behavior, and many other things. So this may result into the showing of or observation of the effect, and then these people, we can say that they are having the disease. So there are many such diseases. Very commonly, genetic diseases are there. Example are muscular dystrophy, familial breast cancer, sporadic breast cancers, lung cancer. So see, you can see in muscle dystrophy, the what is playing maximum role is genetic, then very small role is played by behavior, and very small slightly this one slight role is played by environment. So here we'll see this picture. So here Alzheimer's, Parkinsonism, atherosclerosis, you will see wherever this white portion is more, that indicates that genetics plays very, very important role. And in addition to that, the behavior means own behavior, the behavior with the person, and everything like that. So acquired conditions like trauma, fractures, burn, infection, these are also additional component which can add in, and accordingly they are playing important role. Now this we'll see the major diseases of 21st century. So aging will be one of the problem. The people will be aging faster, and they will have the death rate will be there. Then the diseases will be occurring more at the latter age, and they will be more. So we can see, although there is an increase in the life expectancy, if we'll see that the person in suppose in the beginning of 19th century, the life expectancy was around below 50 age, because many people were dying with infections and all. So average life expectancy was lesser, or there were many childbirth, so average was coming down. Although people were long living, many were having life more than that, but if we'll make the average, because death during delivery, death like that, so it was lesser one. And now we are having the life expectancy is around 80, male female almost same, will parallel one. But here if you will find that in earlier period, the people who are getting the Alzheimer or such kind of genetic diseases, they were lesser comparatively, or chances were lesser. Here we are having more chances. So this is one more problem we are having, that these people will have problems into it. So what are missions of the medicine? Is prevention, diagnosis, and therapy. We know that we call it as a prophylactic use and treatment use. So these are basically diagnostic use. So how we are going to apply the genetics into it, how we will see about it. So here we are having genetics were used as a probe to understand what are the situation, what are the reasons. Then later genes are being used as a factories. During 2000, around 2000 or Y2K we call it as a. So during 2000, genes are being used as a drug, because gene are as a important targets for drug action, and therefore the drastic change are there. Now let us see, we caught as somatic genetic gene therapy. This is it was the background what we have seen. Now use gene as a drug is simply we can call, or basically gene therapy means what? Here wherever the faulty gene were there, we have seen earlier, if you remember, we have taken skin cells, and in that we have inserted during cloning, if you remember. So during cloning it was done that the somatic cells were taken, and this cloning was done. So here somatic gene therapy is there. where whatsoever disorders are there, it is corrected by somatic gene transfer. So somatic gene therapy is used in treatment of hereditary disorder, acquired disorders, some loss of function, or to gain some of the function. These are there. It can be done as a chronic treatment, it can be done as acute treatment, it can be done as a prophylactic or preventive treatment. Now genetic therapy that turn into teenage means it was almost started in 1990. Now almost 20 years are there. Okay, but still we have not found it much effective. Although there are some there in 2002 it was found that some of the protocol were working, but still there are not many gene therapies which are available or which can be made into therapeutics. Now why somatic cells are taken? Because these cells are there. Example, all the cells of the body, we have got germline cell which are making what we have two basic cells. One is somatic cell and germ cells. Germ cell are the cells which are having pluripotent stem cell, we call them, from zygote, because they are having potential to differentiate into many kind of tissues. So therefore, or many kind of cells. So we call them as germline cells. These are coming from the spermatocytes oocyte and their precursor, and upon fertilization they give rise to a descendant organism. So these are germline, and somatic cells are the cells which are all other cells of the body, particularly which are not going into much differentiation, or they are just, but they are viable and growing one. So transformation of germ cell is avoided to exclude the risk of erratic mutations due to insertional mutagenesis into that, and therefore we are not preferring the germ cell line because they are programmed for differentiation. After each cycle they are forming, as we have discussed earlier, that from sperm line, from sperm and from oocyte, the zygote which is formed, which is the first diploid cell, which is single cell, and from that single cell we get trillions of the cell. Many of them are having different functions. So these all different functions they are acquired with time, and these are all because of the differentiation. So this differentiation is more with the germ cell line, and therefore since they are getting changing, these are less preferred. Now how we can use, or where we can use this gene therapy? Where there is no existing cure or treatment possible, which are the diseases, which are the genetic diseases. There are side effects and the limitations of the protein injections are there. Example, in cancer, if we have to use interleukin 12 or some other effects are there, or growth factors we have to use in that in case, and they are toxic or they are causing many adverse effects. So in that cases we cannot use. Then there are many indication for this somatic gene therapy, because it can be a standalone or as a complementary or as a supporting therapy. It is complement to conventional therapy. It increases the specificity of conventional therapy. Then it can cause the increase or specificity of the treatment, and next is it increases the efficacy or of the existing drug. It can improve the quality of life. It can helpful in decreasing the burden of daily injections like insulin like things. So these are that way very important. Then 4 fundamental questions and 4 important things are there. There should be gene transfer should be efficiently occur, because if it is not occurring, we have seen that if you remember Dolly, we were seeing yesterday, that out of 277 such oocytes, only 3 could be successfully this one, and out of that only 1 could implant. So it is not, it takes lot of patience, it takes lot of efforts. So efficiency of gene transfer is very important. Then second thing means whether it is possible. Second is specificity of gene transfer. You know, you have studied genes so far. You know that if gene is not properly inserted, or if the location is changing, then in that case the biggest problem is anything can happen. Then persistence of gene transfer, that is, for that you need persistency, that is, you need a very good tolerance, because and toxicity are there. So again we have to know, you should know that which are the genes, which are the vector, which should be the organ, which type of delivery. These are the things you have to think, that in which disease gene therapy can be useful, which gene has to be taken out, which gene has to be inserted, how much portion has to be taken. Then if this gene is there, what vector is to be used, what should be the target organ, and which type of delivery should be done. So there are various vectors are there, various delivery systems are there, and which can be used in various diseases. So examples of these are, we can divide them into chronic conditions and acute conditions. So these are having slow onset of expression. Here in acute condition, the onset of expression will be fast, it is expected, or it is desired. In chronic condition, we go for initiation of the treatment weeks or months, year before the point of no return. That is, example, we call it as in a cystic fibrosis disease. While here we need it the effect within minutes, hours, or days before the point of no return. That means point of no return means now that has resulted. Once the gene is there, once the gene is expressed, then there is a point of no return, because you cannot turn it back. Persisting expression of the transgene or readministration is required in chronic condition, while here it is not required. Usually based on the compensation of the genetic loss of the function, permanent regain of the function can be there, usually based on augmentation of the resident function. Then regulation of gene expression is often necessary because of the persistence. So you need to control that expression. You can add some factors which are controlling it. Regulation of gene expression is not necessary because it is transient one, it is for short period of time. So these are how the differences are there, how you want gene therapy. Do you want it immediately, or you want it after some time? Anyway, immediate means it still takes hours or days, because it is not that easy. So many things are available, and accordingly we can use it. If we'll compare with the pharmacological, this one, drugs are having molecular weight of 50 to 100 Daltons maximum. If these are protein drugs, the molecular weight can be around 20,000 to 1 lakh Dalton, while here nucleic acids, they are having molecular weight much more. Now you can understand that because of that there are problem of pharmaceuticals, that is how to formulate it and how it can be delivered. So here these classical drugs are synthetically prepared, protein drugs are biologically prepared, nucleic acid they are also biologically prepared. Here these drugs are oral delivery possible. In protein also oral delivery not possible. Here it is oral delivery not at all possible, it is even not thought about. Okay, protein drugs, example insulin. Cellular delivery to act at a cell surface or permit into cell, or like that drugs are there. Here we want that protein should go there and they will act extracellularly. Then it can be delivered as a soluble molecule which are having very smaller size, or nano size rather we call. It can be delivered as a soluble molecules, again nanometer size, but it must be delivered as a complex carrier particle, and it should be bigger one. The treatment of drug is reversible, it is rapidly reversible, even protein is rapidly reversible, but nucleic acid cannot be reversible. So we call it as a point of no return, just above that. So therapy with the nucleic acids, it requires particulated formulation, it is much more complex than previous drug deliveries, and it has different degree of reversibility. So it is not only challenges to the geneticists, but it is also a big challenge to the pharmaceutical people, that is those who are in formulation. They have to make the formulation in such a way that it will not be degraded and it can be doing the job, it will be playing the action at a place where it is required. So three classes of gene delivery is there. We call it as ex vivo, that is outside the body, in vivo, inside the body, topical or in vivo, and another is in vivo systematic delivery. So if you can see that ex vivo means we have applied it outside, and from that it will be taken up. Ex vivo is a topical one, at particular place we have given as a subcutaneous injection, and the third one is known as a systemic delivery, so that it is carried throughout the body. So these are three things. There are various classes of vectors. Now since we have to see that the gene has to reach to place where it is there, so there are various ways by which it is there. Usually they are viral vectors or non-viral vectors. Viral vector are quite popular. See, these are the viruses which are attenuated or which are not harmful. So these viruses are there, in that these DNAs are inserted, and these viruses are made it or they are targeted towards the problem cell areas, the cells where there is a problem. To that area these cells are actually targeted, so that they will reach, the viruses will reach to these cells, and they will able to give the transfection. That is, from that this gene is just like infection. Okay, transfection is that is sort of infection which is comparatively more. It is exposed to 10 to 6 particle per cell in 12 hours. Infection is exposed to 1 particle into 30 minutes. So this much is a more transfection is there, the rate is expected. So that our transfection we call as infection is whole bacteria or virus is there when it is invading. Here transfected means when we are just inserting a gene or any genetic product, we will call it as a transfection. Now most relevant issues are there in the vectorology sector. There are many important issues which are there, vectors, because we want, if we know that we are using virus, and then this virus, if it is itself is a dangerous one, itself is a pathological one, then it can be a problem, or it becomes opportunistic, which was not earlier pathologic, but now it becomes pathogenic. And what human body is doing to that, if human body is acting on it, or human body is causing immune reaction to it. If it is immune reaction, there are 2 3 things. It will destroy the virus. So all your efforts for making gene therapy are useless. Second thing is, because of the immune reaction, the body will start immune reaction, and it can be dangerous one. It can be a very big problem. So we have got this problem that we almost call it as a vectorology. That means you have to study the the goods and pros and cons of various vectors. So these vectors will give you what kind of benefit we have with various vectors. We'll go for next class from this, which are various vectors and what these vectors will be doing in our next class, so that you can have more idea about it. Those who want to study, it was long back available in I think 9th edition of Goodman Gilman. Now Goodman Gilman has removed this gene therapy portion. A whole chapter was there a few years back. Then second thing is there is a small topic